Hello students, this is our chapter Neural Control and Coordination Part 2. Today we'll study about transmission of impulses and second topic is generation and conduction of nerve impulse. So first of all we'll study about transmission of impulses. In transmission of impulses, we'll study about first of all, we will study what is synapse. Synapse is the space between two neurons, the gap between two neurons. So transmission of impulses takes place by two methods. First is electrical synapse and second is chemical synapse. Electrical synapse, there is a continuity between presynaptic and postsynaptic neurons. The continuity is provided by the gap junction between two neurons. The gap junctions are small proteins, tubular structures that allow free movement of ions between the two neurons. Because of this, the action potential reaching the presynaptic terminal produces potential change in the postsynaptic neurons. In electrical synapses, there is minimal synaptic delay because of direct flow of electrical current from one neuron to another. And the chemical synapses, the process of chemical synapse is basically the release of neurotransmitter from the presynaptic neuron, and that causes depolarization in the postsynaptic neuron. So we'll study about this in next slide. Transmission of nerve impulse at a chemical synapse we'll study in this slide. When an impulse arrives at a presynaptic node, calcium ions from the synaptic cleft enter the cytoplasm of the presynaptic node. The calcium ion cause the movement of synaptic vesicles, you can see in the diagram, to the surface of the node. The synaptic vesicles are fused with the presynaptic membrane and get ruptured. This is called exocytosis to discharge their contents. What are the contents? Contents are neurotransmitter into the synaptic cleft. The synaptic vesicles then return to the cytoplasm of the synaptic node where they are refilled with neurotransmitter. The neurotransmitter of synaptic cleft binds to the protein receptors or the postsynaptic membrane. The binding action changes the membrane potential of the postsynaptic membrane, opening channels in the membrane and allowing sodium ions to enter the cell. This causes depolarization and generation of action potential in the postsynaptic membrane. So, having produced a change in the permeability of postsynaptic membrane, the neurotransmitter is immediately lost from the synaptic cleft. So, in the cases, there is neurotransmitter is acetylcholine, which is also called noradrenaline, which is released at the time of nerve impulse. This slide is showing us about chemical synapse and neurotransmitters. Neurotransmitter, as I have told you earlier, acetylcholine is released by all neuromuscular junction between motor neurons and skeletal muscle cells. And one more is noradrenaline, which is also called norepinephrine. Uh, it is secreted by some neurons of synaptic neural system and also by some neurons of central neural system. The nerve cells remain bathed in the extracellular fluid or interstitial fluid containing a large amount of sodium chloride and bicarbonates. In addition, it contains nutrients and oxygen for supplying to the cell and carbon dioxide and other metabolic waste released into it by the body cells. So, the theory was proposed by English neurophysiologist Hodgkin and Huxley in 1930. The theory states that electrical events in the nerve fiber are governed by a differential permeability of its membrane to sodium and potassium ions, and that these permeabilities are regulated by an electric field across the membrane. According to this theory, the process of nerve impulse conduction is divisible into two main phases. First is resting membrane potential of nerve and action membrane potential of nerve. We'll learn about this in the next slides. Now we'll study about some terms. First of all, we'll study about potential difference and then membrane permeability, resting potential difference, and sodium potassium pump and polarization. First of all, we'll see the electrical potential difference across the resting plasma membrane is called resting potential. The state of resting membrane is called polarized membrane. And the electrical potential difference across the plasma membrane at the site A is called action potential, which is in fact termed as the nerve impulse. And sodium potassium pump is present. So as I have already told you that there are sodium ions and potassium ions present, the outgoing and ingoing of potassium ions manage the protein gates and that use energy to transport sodium ions out and potassium ions in. Let's see how it happens. Polarization, the situation when the membrane separates a more negative inside from the positive outside. In this slide, we can see polarized membrane that is in resting potential. Uh, in a resting nerve fiber means a nerve fiber that is not conducting an impulse. The exoplasm is neuroplasm of axon. Inside the axon contains high concentration of potassium ions and negatively charged proteins and low concentration of sodium ions. In contrast, the fluid outside axon contains a low concentration of potassium ions and a high concentration of sodium and thus form a concentration gradient. These ionic gradients across the resting membrane are maintained by active transport of ions by sodium-potassium pump, which transports 3 sodium ions outwards and 2 potassium ions inwards. As a result, the outer surface of axonal membrane possesses a positive charge, while the inner surface becomes negatively charged. The electrical potential difference across the resting plasma membrane is called as resting potential, and the resting membrane is called polarized state. Now next is depolarization. When a stimulus of adequate strength means threshold stimulus is applied at site A, for example, point A on the polarized membrane, the membrane at site A becomes freely permeable to sodium ions. This leads to a rapid influx of sodium followed by a reversal of polarity at that site. That is, the outer surface of membrane becomes negatively charged and the inner side becomes positively charged. The polarity of the membrane at the site A is thus reversed and hence depolarized. The electrical potential difference across the plasma membrane at the site A is called action potential, which in fact is termed as nerve impulse. At sites immediately ahead the axon, for example site B, membrane has a positive charge on the outer surface and negative charge on the inner surface. As a result, current flows on the inner surface from site A to site B. On the outer surface, current flows from site B to site A to complete the circuit of current flow. Hence, polarity at the site is reversed and an action potential is generated at site B. So likewise, the sequence is repeated along the length of axon and consequently the impulse is conducted. The rise in the stimulus induced permeability to sodium ions is extremely short-lived. This is quickly followed by a rise in permeability to potassium ions. Within a fraction of a second, potassium ions diffuse outside the membrane and restores the resting potential of the membrane at the site of excitation and is called repolarization, and the fiber becomes once more responsive to further stimulation. In this slide, I have given some easy definitions: depolarization, action potential, refractory period, saltatory conduction, and all or nothing response. So here one word is new for you: saltatory conduction of nerve impulse. The properties of impulse conduction described so far apply to unmyelinated neurons. However, the myelin sheet of many neurons in the body insulates those axons except at the nodes of Ranvier. When an impulse travels along a myelinated neuron, depolarization occurs only at the nodes. It leaps over the myelin sheet from one node to next. This process is called saltatory conduction. So saltatory conduction means it jumps from one node of Ranvier to another. It is faster than in unmyelinated fibers.